Today, we are going to talk about photorespiration. Photorespiration or C2 cycle is a metabolic repair system in plants. It is discovered by Diker and Dio in 1959. Photorespiration refers to a process in plant metabolism where the enzyme RuBisCO oxygenates ribulose 1,5-bisphosphate and wasting some of the energy produced by photosynthesis. It enables the carbon dioxide fixation enzyme RuBisCO to sustainably operate in the presence of oxygen. Carbon fixation results in the formation of two molecules of phosphoglycolate, means 3-PGA, that are integrated into the Calvin cycle, ultimately to form sugars in the presence of RuBisCO. So RuBisCO is a bifunctional enzyme that catalyzes not only ribulose bisphosphate carboxylation but also oxygenation. Ribulose bisphosphate oxygenation generates one molecule of PGA and one molecule of 2-phosphoglycolate. Because this is the first product of photorespiration, phosphoglycolate, it's a 2-carbon molecule which is later converted to glycolate. So that's why photorespiration is also called C2 cycle. 2-phosphoglycolate is a toxic compound to many enzymes in photosynthetic metabolism and oxidizes organic carbon without seemingly generating ATP. ATP, energy-rich molecules. So photorespiration is generally considered a wasteful process. On the other hand, photorespiratory pathway also rescue 3/4 of the carbon in phosphoglycolate that would be otherwise inaccessible for further metabolism. Thus, photorespiration can also be regarded as an important pathway that makes the best of a bad situation caused by RuBisCO's seemingly inevitable oxygenase activity. First of all, we'll start with the enunciation of photorespiration. In C3 plants, the first step of photosynthesis is the fixation of carbon dioxide. Now you can see here, this is chloroplast and this is C3 cycle, Calvin cycle. So in C3 plants, first step of photosynthesis is the fixation of carbon dioxide by ribulose bisphosphate. For every molecule of carbon dioxide fixed, this reaction produces two molecules of 3-carbon acid, that is 3-phosphoglycerate, 3-PGA, in the presence of enzyme RuBisCO. Now photorespiration. Photorespiration is initiated by oxygenase activity of RuBisCO. Oxygenase activity, here addition of oxygen, right? So photorespiration is initiated by oxygenase activity of enzyme ribulose 1,5-bisphosphate carboxylase oxygenates. Photorespiration is higher under hot and dry conditions, mainly for 3 reasons. First is the specificity of RuBisCO for carbon dioxide relative to oxygen decreases with temperature. Second is the solubility of oxygen in aqueous solution such as cytoplasm and the chloroplast is less affected by high temperature than the solubility of carbon dioxide. Third is plants exposed to limited water supply reduce carbon exchange in order to minimize evaporation. So carbon dioxide concentration around RuBisCO rapidly decrease where oxygen is available in excess. This favors the oxygenase over the carboxylase reaction. Photorespiratory pathway. Photorespiration involves an interaction of 3 organelles: chloroplast, peroxisome, and mitochondria. When temperature and oxygen content are high, the affinity of ribulose bisphosphate carboxylase increases for oxygen but decreases for carbon dioxide, right? It converts ribulose bisphosphate to 3-carbon compound, means 3-PGA and 2-carbon compound phosphoglycolate. Now, from chloroplast, glycolate is diffused to peroxisome, where it is oxidized to glyoxylate This is glyoxylate. Here glyoxylate is used to form amino acid glycine. Now glycine enters mitochondria, where two glycine molecules, means 4 carbons, give rise to one molecule of serine and one molecule of carbon dioxide. Here 1 carbon. Now this serine is taken up by peroxisome and through a series of reaction is being converted into glycerate. This is glycerate. This glycerate now leaves the peroxisome and enters the chloroplast, where it is phosphorylated to form PGA. PGA molecule enters the Calvin cycle and to make carbohydrates, but one carbon dioxide molecule is released here in mitochondria during photorespiration, has to be refixed. Now, positive and negative effects of photorespiration. First of all, we'll start with the negative effects of photorespiration. Photorespiration is generally considered a futile cycle, as I already told you, that potentially decreases photosynthetic carbon fixation by more than 25%. I'll repeat here again. Final product of photorespiratory cycle is one molecule of 3 carbon compound, means 3 PGA, and one molecule of 2 carbon compound, phosphoglycolate. Now, PGA molecules enter the Calvin cycle to make carbohydrates, but one carbon dioxide molecule released in mitochondria. This means 75% of the carbon lost by oxygenation of ribulose bisphosphate is recovered and 25% is lost as a release of one molecule of carbon dioxide. Phosphoglycolate 2-PG is a toxic compound. Another negative effect, too many enzymes in photosynthetic metabolism and oxidizes organic carbon without generating ATP. So photorespiration is considered as a wasteful process. One important thing I would like to discuss with you, carboxylation pathway uses 3 ATP and 2 NADPH per ribulose bisphosphate regenerated and produced a carbon in hexose, whereas oxygenation pathway of photorespiration uses 3.5 ATP and 2 NADPH per ribulose 1,5 bisphosphate regenerated with no addition of organic carbon. Now, positive effects of photorespiration. Photorespiration removes toxic metabolic intermediates. Second, it protects from photoinhibition. Then it supports plant defense reaction. It also rescues 3/4 of the carbon in phosphoglycolate that would be otherwise inaccessible for further metabolism. Now, after watching this video, you will be able to answer these questions. So, after watching this video, you can try to answer these important questions. Thanks for watching. If you like this video, make sure to like and subscribe for more. Thank you.